Okay, hello to all of you. Let's discuss chapter number 21. And this chapter is about lipid metabolism. So in this we are going to talk about, um, first of all, we are going to talk about lipid catabolism, and then we are going to talk about the biosynthesis of the lipids. So, so basically lipids, as you know, they have the very high energy in them, and they are the macromolecules which have the highest energy in them. So they represent an efficient way of storing chemical energy, and the oxidation of these lipids release large quantities of energy through the production of acetyl coenzyme A, NADH, and FADH2. So you have already studied, we have already studied the metabolism of carbohydrates. Uh, we have talked about glycolysis. We have talked about Krebs cycle. We have talked about the conversion step, electron transport chain, and oxidative phosphorylation. So you have seen that in the second step, acetyl coenzyme A is made, and then that enters into the Krebs cycle, and then the energy, which is in the form of high energy electrons, it's taken by the electron carriers like NAD plus and FAD, and they all taken to the inner membrane of mitochondria. So energy is basically in the form of all these molecules. So in the lipids, we have basically triacyl glycerol, which is also called triglycerides, or phosphoacyl glycerol, which are phospholipids. So triacyl glycerol, it's a main form of chemical energy, and phospholipids or phosphoacyl glycerol, they are present in the membranes. And the triglycerides or triacyl glycerol, it can be hydrolyzed by using lipases, and the phospholipids can be hydrolyzed by using phospholipases. And this basically shows how the how the fatty acids, they are separated from the or liberated from the triglycerides or triacyl glycerols, and then these fatty acids can undergo the process of beta oxidation. So this shows that there is a receptor in the plasma membrane. Receptors are usually the proteins. So the hormone comes and binds on the receptor, and then as a result of that, so this is all basically signal transduction pathways. Then as a result of that, there is an enzyme called adenyl cyclase. It gets activated, and this enzyme adenyl cyclase can convert ATP into AMP, cyclic AMP. This is called C is for cyclic, C word is for cyclic, AMP is adenosine monophosphate. So this enzyme adenyl cyclase converts ATP into cyclic AMP, and then this cyclic AMP can activate another protein kinase, and that protein kinase, when it gets activated, it can activate triacyl glycerol lipase. This is another enzyme. So these are like a cascade of reactions that this enzyme gets activated. Then there's a production of cyclic AMP. Cyclic AMP is a secondary metabolite, secondary messenger basically, and then the cyclic AMP activates another kinase. Then the kinase is going to activate this lipase, and the function of this lipase is so it's a triacyl glycerol lipase. So this enzyme can help in removing one of the fatty acid from the triacyl glycerol, and then this is the molecule with two fatty acids. So this is called diacyl glycerol. So this is glycerol with two fatty acids, diacyl glycerol. Then with the help of the another enzyme called DAG lipase, the second fatty acid is removed, and now there is glycerol with one fatty acid. It's called monoacyl glycerol. It's called monoacyl glycerol. And this from monoacyl glycerol, with the help of the enzyme monoacyl glycerol lipase, the last fatty acid is also liberated, and it results in the formation of fatty acid and a glycerol, and then these fatty acids can enter into beta oxidation. So now we are going to talk about how these fatty acids, they are oxidized, which means how energy is released from them. So first of all, the fatty acid oxidation begins with the activation of the molecule. Okay, so the very first thing which happens in the uh, fatty acid oxidation is the fatty acid activation. So how it is activated? A thioester bond between carboxyl group of fatty acids. If you remember what fatty acids are, the long chains of carbons and hydrogens with a carboxyl end at the end. So a thioester bond is formed between the carboxyl group of the fatty acid and the thiol group of the coenzyme A. Okay, so between fatty acids and the coenzyme a thioester bond is formed, and a thioester bond is formed between the carboxyl group of the fatty acid and the thiol group of the coenzyme A. And this takes place with the help of the enzyme called acyl coenzyme synthetase. So acyl coenzyme A synthetase, this is the name of the enzyme. It catalyzes the formation of ester bond, and it requires ATP for its function. And this bond formation, which is thioester bond formation, or it's also called esterification, takes place in the cytosol, but the rest of the reactions of fatty acid oxidation occur in the mitochondrial matrix. And acyl coenzyme A can cross the outer mitochondrial membrane but not the inner one. And then we are going to focus how they enter into the mitochondrial intermembrane, and from intermembrane they reach the mitochondrial matrix. So this basically shows you the esterification reaction. So this is the fatty acid, this is the carboxyl group, this is the R group, which means this is long chain of hydrogens and carbons. So first of all, there's an intermediate formation with the help of the ATP. So the fatty acid forms a bond with AMP, and there's a release of two phosphates. So the intermediate which is formed is called acyl adenyl intermediate, and then it reacts with the coenzyme A and results in the formation of the thioester bond. So this is activated acyl group, and this is the activation of the fatty acids. So the fatty acids, they are linked with the coenzyme A. So this takes place with the help of the enzyme acetyl coenzyme A synthetase. So so now the acyl coenzyme A is formed, and then it can enter so this acetyl coenzyme A can enter from, so this is the cytosol. Okay, so you can see here esterification takes place in the cytosol. So if you look at this picture, this is the cytosol. Here esterification has taken place, which means the linking of fatty acid with the coenzyme A. So this has taken place here, and this can enter from cytosol to the intermembrane space. So this is mitochondrial outer membrane, this is mitochondrial inner membrane, this is the intermembrane space. So this acyl coenzyme A can enter from cytosol to intermembrane space through the outer membrane. Okay, but in the intermembrane space, this acyl coenzyme A, it actually gets conjugated with a molecule called carnitine. Okay, it gets conjugated with carnitine, and then this complex is formed, this compound is formed, and this coenzyme A is released. It's called acyl carnitine. Okay, and then this acyl carnitine can enter into through the inner membrane, it can enter into the mitochondrial matrix, and the formation of, so the formation of acyl carnitine, it takes place again with the help of the enzyme called carnitine acyl transferase. Okay, and then in the matrix, from this compound acyl carnitine will be released, and coenzyme will be also, and then the fatty acid will be entering into the beta oxidation. Oh, I'm sorry. Coenzyme A is actually added because coenzyme A was released here. It's liberated here, and then this was getting attached to the carnitine. So there is no coenzyme A here. So it goes from intermembrane space to the mitochondrial matrix, and then carnitine is separated. You can see here, and then this uh, fatty acid part is combined with coenzyme A again, and then this part enters into the beta oxidation process. So there is more info, whatever I said, it's written here. Let's quickly go over it. So acyl group is transferred to carnitine. And what is carnitine? It's a molecule used in fatty acid metabolism to shuttle acyl groups across the inner mitochondrial membrane. And the trans-esterification reactions are catalyzed by the enzyme called carnitine acyl transferase, which transfers the fatty acyl group to the carnitine. So that is what is happening here. So what is happening here? So this group is getting combined with this carnitine, and this compound is formed. So this is taking place with the help of this enzyme called carnitine acyl transferase, and there are two kinds of these enzymes called carnitine palmitoyl transferase CPT1, which is found on the cytosol side of the inner mitochondrial membrane. So you can see here cytosolic side of the inner mitochondrial membrane is this. So this is that enzyme, and then this is the second enzyme carnitine palmitoyl transferase 2, which is found in the mitochondrial matrix. So this is that enzyme. Okay, now after that what happens to this compound? It goes through beta oxidation. Now beta oxidation has 4 steps, which you need to understand what those 4 steps are. So repeated series of reactions. So in beta oxidation what happens is it involves 4 different steps, and in those steps what happens, every cycle cleaves 2 carbon units from the carboxyl end of the fatty acid. It cleaves 2 carbon units from the carboxyl end of the fatty acid. So let's see what happens here. So you can see here, this is your fatty acyl coenzyme A. In the first step, this is called oxidation. So which means in this step hydrogens are removed, and it takes place with the help of the enzyme called acyl coenzyme A dehydrogenase. And the hydrogens which are removed, they are taken by FAD, and FAD is converted into FADH2. So the hydrogens are removed. So these are your carbons, okay, from which these hydrogens are removed, and when these hydrogens are removed, it will result in the formation of double bond here, okay. And then this compound is called trans enol coenzyme A, okay. And in the next step, this compound which is formed from this, and there's a formation of double bond here, the next step is like a hydration reaction in which water is added. That takes place with the help of the enzyme called enoyl coenzyme hydratase. So it's all on the basis of what is happening in the reaction. The reaction is called hydration reaction. So which means to this compound water will be added, which is H2O. So you can see here it's all highlighted here. So this is your double bond here. So because now the water will be added, this double bond will be broken, and you can see one H is added on this carbon and OH is added on this carbon. That results in the formation of this compound. And then the third step, again there's the oxidation. It takes place with the help of this enzyme L-hydroxy acyl coenzyme A dehydrogenase. So it involves the removal of hydrogen atoms, and so this is oxidation step. In this NAD, so in this step NAD plus gets converted into NADH. In this step FAD is converted into FADH2. Here NAD is converted into NADH. So here again hydrogens are going to be removed. So you can look here at this part. Again the hydrogens are removed. So if you look here, This is your carbon. Okay, so this one hydrogen is removed from here, another hydrogen is removed from here, so this carbon is left with oxygen. So this, you can see, this carbon is left with oxygen. So this is the compound which is formed, which is called beta ketonyl coenzyme A. And then in the fourth step, which is a kind of a cleavage step, which involves the enzyme called thiolase. In this, cleavage takes place, and the cleavage takes place here. Okay, the cleavage takes place here, and there's also addition of one coenzyme A. So which means cleavage means the bond is broken here. So this part gets separated, this part gets separated. So you can see here, if you look here, so this part is this. Okay, so there is this coenzyme A, carbon with oxygen, then you have CH2, okay, CH2, and the R group is here, and this part gets separated, which is your this part, and there's also addition of this, so that's why you get this kind of a compound. So there is this is the carbon, this is this carbon, then this CH2. So because the this is getting broken from by the cleavage takes place here, so this is this part. Okay, so carbon with oxygen, then this carbon with two hydrogens, and instead of R it's hydrogen because the R is with this side. So this is the part which separates. So in this way, in the beta oxidation, in every cycle of these four reactions, two carbons will be removed. So this is just showing, so in every step of beta oxidation, in every cycle of the beta oxidation, two carbons are removed. So stearic acid, which has 18 carbons, give rise to 9 2 carbon units after 8 cycles of beta oxidation. Okay. So this is basically, you can go over it, it just says what is the yield after the oxidation of fatty acids. So the energy released by the oxidation of acetyl coenzyme A formed by beta oxidation fatty acids can be used to produce ATP. 8 cycles of beta oxidation are required for the oxidation of 1 mole of stearic acid to 9 moles of acetyl coenzyme A. This is all the complete reactions written. So you can go over it. I'm not going to talk. Okay, so this was all beta oxidation. So in the fatty acid catabolism, beta oxidation is very important. The next is fatty acid biosynthesis, how fatty acids they are made. So anabolism is not the exact reversal of the reactions of beta oxidation. So similarly, if you remember, gluconeogenesis is also not the exact of glycolysis. There are some steps same, but some are different. So fatty acid biosynthesis means they are anabolic reactions. They occur in the cytosol. So acetyl coenzyme A, it can be formed either by the beta oxidation of fatty acids or by the decarboxylation of pyruvate. If you remember, pyruvate gets converted into acetyl coenzyme A, and this acetyl coenzyme A, it condenses with oxaloacetate to form citrate. So in the fatty acid biosynthesis, the first step is the carboxylation of acetyl coenzyme A in the cytosol. Okay, and this takes place, so there's a carboxylation of acetyl coenzyme A in the cytosol, and this takes place with the help of the enzyme called acetyl coenzyme A carboxylase, and this enzyme requires manganese, biotin and ATP. And what is biotin? It's a carrier of carboxyl group and an important cofactor in the reaction. And when the acetyl coenzyme A is carboxylated, it produces a key intermediate called malonyl coenzyme A. And you will see that acetyl coenzyme A and malonyl coenzyme A, they will be like the building blocks for the making of the fatty acids. So this is your acetyl coenzyme A. So it reacts with, it needs ATP, and this is the bicarbonate. This is for the carboxylation reaction. It takes place with the help of the enzyme called acetyl coenzyme A carboxylase and biotin and manganese. So as a result of the carboxylation of acetyl coenzyme A, malonyl coenzyme A is formed. So this basically shows you uh, the first part. It shows you um, the acetyl coenzyme A carboxylase reaction. This is malonyl coenzyme A for the fatty acid synthesis. So this is showing you how from acetyl coenzyme A it gets carboxylated and malonyl coenzyme A is formed, which is required for the formation of the fatty acid synthesis. And this is basically showing you the mechanism for the acetyl coenzyme carboxylation reaction. The bicarbonate, this one, is activated for carboxylation reactions by the formation of N-carboxy biotin. Okay, N-carboxy biotin. So this is N-carboxy biotin, and how it is formed. So in this reaction is driven by ATP. ATP drives the reaction forward. So ATP drives forward with the transient formation of carboxyl phosphate intermediate. So you can see here. So there is a formation. So this reaction is driven by ATP, and it helps in the formation of carboxyl phosphate intermediate. Okay, and then the biotin combines here. You can see like, and in this step, so these are actually the mechanism of this reaction. And in this part, what is happening is the acetyl coenzyme A carb anion. This is what it is. It attacks on the carboxyl carbon. This is the carboxyl carbon of N-carboxy biotin. Okay, so this is what it is, which was formed here. So this carboxyl carbon of N-carboxy biotin, okay, and it yields the carboxylated product, and biotin is free. And this carboxylated product is the malonyl coenzyme A. Okay, and acetyl coenzyme A and the malonyl coenzyme A, they are basically the building blocks for the formation of the fatty acids. Okay, so this is showing you basically, you don't need to know in a lot of details. You just need to know that how so it's the way you have seen in the oxidation that the two carbons, in every step 2 carbons are removed. Similarly, in the fatty acid biosynthesis, 2 carbons will be added in every step. So here you can see this is the acetyl coenzyme A and this is the malonyl coenzyme A. They act as the building blocks. They act as building blocks for the synthesis of fatty acids, and they are introduced as acyl carrier protein conjugates. So all the steps are taking place here, and we don't need to go into that many details. So they are getting basically they are getting conjugated with some carriers, and these acetyl and the malonyl coenzyme A, they are basically used as acyl carrier protein conjugates, and as a result of that, decarboxylation takes place. You can see here carbon dioxide being released here, and the decarboxylation drives the enzyme called beta ketoacyl ACP synthetase, and it results in the addition of 2 carbons to the growing chain. And here there are some reactions taking place which are actually the reverse of what is taking place in the beta oxidation. So let me get it big. So here you can see this is acetoacyl ACP. Here actually reduction is taking place, which means NADPH is actually giving the hydrogens. So this is reduction reaction, and it's taking place with the help of this enzyme. So the hydrogens are being added in this step. One is added here, another is added here, And then the next step is hydration reaction. Um, it's also opposite to the reaction which is taking place in beta oxidation. In the beta oxidation, the second step is actually hydration. Here is dehydration, and here again the next step is the reduction, which means the addition of the fatty acids. Okay, so you can go with this. Okay, synthesis of acyl glycerols and compound lipids. So lipids such as, so we just talked about how fatty acids are made, biosynthesis of fatty acids. Now the next step is lipids such as triacyl glycerols, which means triglycerides, phosphoacyl glycerols, phospholipids and steroids. They are actually derived from the fatty acids and the metabolites of fatty acids. And the glycerol portion of lipids, the glycerol portion of the lipids is derived from glycerol 3 phosphate. So now let's see how they are made. This is one of the pathways for the biosynthesis of triglycerides. So you can see the glycerol 3 phosphate, because these triglycerides, they have glycerol in them. So this glycerol can be either made by with the help of glycerol getting converted to glycerol 3 phosphate with the help of the enzyme called glycerol kinase, or it can also be derived from the product of glycolysis called dihydroxy acetone phosphate. It can also with the help of the enzyme glycerol 5 phosphate dehydrogenase, it can be converted into glycerol 3 phosphate, and then with the help of the enzyme called glycerol 3 phosphate O-acyl transferase, the this acyl coenzyme A group is added. Sorry, the acyl group is added, not the coenzyme. So you can see here, this is the enzyme called glycerol 3 phosphate acyl transferase, and it helps in the transfer of the acyl group to the glycerol 3 phosphate. So here you can see you can see here the addition of the acyl group here, and what is formed, the compound formed is called lysophosphatide. It's called lysophosphatidate. And then in the next step, again with the help of the same enzyme called glycerol 3 phosphate acyl transferase, then again the acyl group is added to the another OH group of the glycerol, and the compound which is formed is called phosphatidate. And then with the help of the enzyme called phosphatidate phosphatase, the phosphate group is removed, and then what is formed is called 1,2-diacyl glycerol, and then with the help of the enzyme called diacyl glycerol acetyl transferase, again the acyl group is, the third acyl group is attached, and as a result of that, a triglyceride is formed. So that's all what you need to know. If you have any question, please let me know.